Hi, a very good morning or good afternoon to all of you. I'm Ivan, the Scientific Affairs Manager of Sebia APAC. Today we will be talking about immunotyping, and I really hope that at the end of this session, you will see the whole results interpretation of aspects of immunotyping simplified for your practical application. I'll welcome you to post your questions in the questions chat box along the way. And should you have any other questions beyond this webinar, you're warmly welcome to send these questions to me. I pasted my email in the chat box and I've also included the survey link as well. And I would really hope that you could participate in the survey link to help us improve our webinars, our future webinars. So in a normal person, the plasma cell composition will be normal and you will be producing antibodies in equal amounts, whichever antibody clones that are present in the body. And we know that this is a huge repertoire of antibodies. There's no abnormality and everything is just produced in normal amounts. And what results is that you will see in the gamma fraction of the serum protein electrophoresis, a Gaussian aspect of the gamma curve. Okay, so one way to remember this is that it's like a normal distribution, statistical distribution, because all the antibodies in the body, right, are in the right amounts. Okay. However, this normal distribution would be skewed and affected in the case of a malignant situation like multiple myeloma. Okay, it may not be restricted to multiple myeloma. Some other malignant conditions like Waldenstrom's macroglobulinemia, it also produces abnormal monoclonal proteins in larger amounts. When this happens, because one plasma cells that becomes malignant will overgrow. Sometimes it will also displace the other normal plasma cells in the bone marrow and reducing the amount of antibodies produced by the normal cells. In such situations, we will even see immunoparesis happen, like the normal antibodies that are produced are reduced significantly. And what we will see is a sharp peak of a monoclonal antibody because cancer is a clonal condition. Usually it starts with one cancer cell becoming disordered and then it will produce the antibodies in large amounts. Hence, you see that sharp peak. So after getting the serum protein electrophoresis and seeing the monoclonal antibodies and protein present, what would be the next step? Importantly, you have to identify what that isotype of the antibody is. So is it an IgG, is it AM, D or E? Because the nature of the antibody isotype will tell you a very different prognosis for each of the different cancer types. Okay, so and also importantly, what would be the light chain composition of those antibodies? So there are two methods to identify the isotypes. One would definitely be the conventional immunofixation method using agarose gel electrophoresis. But today our focus will be on immunotyping, which uses capillary electrophoresis. Okay, so with the capillary electrophoresis instrument setup, you can be assured that this process has a better workflow and increased automation and traceability compared to the conventional gel method where you still have to perform the individual pipettings. Okay, of course, similar with the immunofixation concept would also be that anti-serial, the sample will be mixed with anti-serial of different specificities. And this will target all the different heavy chains. The main heavy chains that we look at will be IgG, IgA, and IgM. The same goes for immunofixation, that you also screen these antibodies first. Why? Because they are the antibodies that present in larger amounts. All right, so we always screen those that most frequently would happen. And should there be any exceptions, there are the exceptional anti-serials to look at other scenarios like IgD and IgE, right? Or free light chains. Okay. So how does the immunotyping principle works? If you look at immunofixation in the gel, you are using the anti-serum to react with the antibodies that are present in the gel. And after this reaction, the anti-serum antibody complex will stay on in the gel. That's why we call it fixation. They are fixed within the gel and you're not able to wash them away. However, immunotyping employs a different mechanism. What it does, even though with the anti-serum, so the anti-serum is represented here in red as highly negatively charged antibodies. So some modification has been done to this anti-serum reagent. And when you mix it with the patient's sample, which is denoted as the green highlighted immunoglobulin within the serum, I want you to know that the anti-serum is blind. It will always bind to the isotype, whichever isotype that it targets. Like for example, if it binds with the patient's IgG, you bind every IgG, whether is it monoclonal or is it polyclonal, all right? So when that complex forms within capillary electrophoresis, it becomes a highly negatively charged complex. So we know that in capillary electrophoresis, we are migrating our samples throughout a very fine capillary. So at the end of the capillary will be the detection, and all the species that are going through the detector will be detected, and it gives you the display of the serum protein electrophoresis curve. Okay. However, because the anti-serum and the antibody complex is highly negatively charged, when they are run through the capillary electrophoresis, they will migrate further away, even past the albumin portion, okay, on the anodic site. So what you see like now is that the antibody complex, anti-serum antibody complex is far away out of the albumin curve. And if you look at the residual curves when interpreting the immunotyping results, you always see this on the left side of albumin, all right, which means that your sample has been immunosubtracted, right? So these are telltale signs that you can always understand these results and profiles when you look at them. So essentially, immunosubtraction has happened. So when you look at the literature and look at immunotyping, you might look at an interchangeable word that's used, which is immunosubtraction. So these are actually the same things, okay? If you're searching through the literature, know that it is the same as immunotyping or IT, that we call it here. Right, so what is the strength of immunotyping would be that we know that capillary electrophoresis, it has superior resolution compared to many other charge separation methods. And also in comparison to gel electrophoresis, for example, the higher resolution will allow you to separate your fractions even more, which means that if two monoclonal proteins are migrating closely together, there's a higher chance that they will be separated apart for you to identify them as a biclonal issue, okay, in capillary electrophoresis. Okay, so it also removes away by immunosubtraction backgrounds that are very strong. For example, the polyclonal background of IgG, okay, with the same principle that I just explained to you. So now looking at the normal serum protein electrophoresis profile, we always need to look at the normal profile to know what is abnormal. Okay, so I would like to remember the normal profile when I look at the albumin is always high, and then after that it proceeds like going up a curve here and then coming down. Okay, so alpha 1 goes up to alpha 2, kind of goes up to beta 1 in terms of the height, and then it goes down to beta 2. Okay, so you know that alpha 2 is always higher than alpha 1, and beta 1 is always higher than beta 2, having the picture over here in your mind. Okay. So there should not be any distortions or extra peaks compared to what is normal here in this fraction. And when you look at the gamma fraction, it should remain in the statistically normal manner of the Gaussian aspect of the gamma curve. Okay. So what are some scenarios where you see abnormalities? If you look at the far right panel that's in this slide, this is the very idealistic and typical monoclonal peak in gamma. Okay, a very strong peak on the right side. However, the monoclonality may present itself in other ways. Okay, so if you look at the additional peak in alpha 2, it can happen as an additional peak in beta 1 or beta 2. Why? Because the superior resolution of capillary electrophoresis allows better separation. You're more likely going to detect abnormality in this way. Okay, so knowing the normal profiles, these will be abnormalities. And if you extend it to look at the, for example, an isolated increase in alpha 2. Alpha 2 is like a acute phase protein. You're supposed to see it when there's elevated inflammation. But however, if there's no elevated inflammation or any nephrotic context, and you see that increase in alpha 2, it should also infer that something could be happening in multiple myeloma that's related. All right. Of course, there can be inflammation in that multiple myeloma as well. So likewise, the same goes for the isolated increase in beta 1, when you do not have an underlying iron deficiency or hemolysis condition that will leak out contents from a red blood cell that might contribute to this peak. Okay, it could be a multiple myeloma underlying it as well, or monoclonality. Okay, and as we said, beta 2 should be lower than beta 1, and if you see that it's higher than beta 1 without any inflammation or hepatitic context, it should ring a bell about possible monoclonalities as well. Okay, and not always the case. If the monoclonal peak is in the gamma portion, you will see a very idealistic, huge monoclonal peak. It really depends on the stage of the disease, and this gives a lot of visibility to identify monoclonality earlier as well, even when their levels are not as high. In this situation, you see a distortion in the gamma fraction. Okay, so even this little distortion that disrupts the Gaussian curve, right, tells you much about possibility of the monoclonality, which warrants for further investigation with immunotyping to understand what that isotype is. So now let us look at how do we go about to systematically interpret an immunotyping result. Okay, although it looks like there are many panels that are present here, 6 panels, the curve that's in yellow on the top left-hand side is also called the reference curve. Okay, so this is the sample that is not reacted to the anti-serial. There is no immunosubtraction. Okay, if you look at the left side of urine, there's nothing. So there's no immunosubtraction to these. We call it the reference curve because we constantly take reference back to it when we are comparing it to the other frames. All right. So then we look at the other frames, and importantly, so one, we are looking at the reference curve. We have to identify, we always identify from the reference curve where the abnormality is. In this case, it's rather obvious. It's in the monoclonal peak in the gamma fraction. So we will go back to look at the other panels, back at that same abnormality to see which one of them has been subtracted in the residual curves. The residual curves will be the overlaid curves in blue that you see in the other panels. So they identify, and in this case, it's rather apparent that the subtraction is only happening in IgG and in the lambda window. All right, so from this combination, this disappearance of the abnormality, we can be sure that this is an IgG lambda monoclonal band. Okay. The additional advantage of having your immunotyping being more digital on a capillary electrophoresis platform is also that you are able to zoom into your curves. All right, because some of the distortions may be too small for you to detect it by just looking at the smaller magnifications. All right, so you're able to increase it for the various amounts of zoom, like in this example, a 4x zoom at the gamma portion where you see the monoclonal peak. All right, and looking carefully at that monoclonal peak is actually like an extension to the Gaussian aspect of the gamma curve. All right, so in this case, where does the subtraction happen? It happens in the IgG and IgG lambda, right? So likewise, this is a IgG lambda. Okay. So now looking back at the normal serum protein electrophoresis curve, So what if we are to subject this normal serum sample for immunotyping? Would there be any form of subtraction that happens? So this will actually help us understand how immunotyping works even better, and what are the composition of antibodies that are present in the body. Okay, so once again, we zoom into the beta 1 to gamma region of the curve. Right, there's no abnormality here, but we do see some form of subtraction happening here. So remember what I told you, the anti-serial is blind. It actually targets all the antibody isotypes, regardless of whether is it a monoclonal protein or is it a normal protein as part of the polyclonal repertoire. All right, so we can see that in the IgG window, there's about a huge subtraction. The reason is because most of the antibodies that are present within our polyclonal mixture is IgG. Okay, 80% of it is IgG. Then the next populated antibody will be IgA, which takes up about 15%. But the important thing to note is that for IgA, the presence of IgA is from the end of beta 1 to about the mid of gamma region. So this is the span of the region where you could see potentially where IgA is. And in this case, because it's a polyclonal IgA, you can see a spread of that. We call it like a bridge aspect from beta 1 to the mid of gamma. All right. And if you look at the next populated antibody, which is IgM, it is much lower. It's about 5%. And the presence of it always spans at the left side of the gamma fraction. Okay, so it's important to remember these localized positions. Okay, and then if you look at the light chain compositions, remember, because most of your antibodies are the IgGs, so the representation of these light chains, 2/3 kappa and 1/3 lambda subtraction, is also because of the IgG. That is the highest amount of antibody that's present in the polyclonal mixture. Okay, so in the polyclonal mixture of all antibodies, having IgG as the most amounts, 2/3 of the antibodies will be kappa, 1/3 will be lambda, right? So how do you make a difference between a polyclonal increase and a monoclonal increase? A polyclonal increase always have a white base and a rounded top. Okay, so when you see that in your immunotyping and there is subtraction in the kappa and the lambda, as you will see later, it's always a partial subtraction. So just now we talked about 2/3 kappa subtraction, 1/3 lambda subtraction. That is an example of a partial subtraction, which means both of the light chains are partially subtracted. If you look at the monoclonal situation, the monoclonal peak always have a narrow band and a pointed top. And when the subtraction happens, it is not a partial subtraction, but a complete subtraction of either of the light chain types. Why? Because the monoclonal protein only has one light chain identity, right? If it's an IgG kappa, it can't be a lambda as well, unless you have two clones. Okay, so that explains the subtraction difference between them. Okay, so now in this polyclonal increase situation in IgG, once again, a white base and a rounded top. Okay, so identify where the abnormality is in the reference curve. Okay, and now we zoom in, all right, and then we will look at the other windows to see where is the abnormality completely subtracted. It is completely subtracted in IgG, so we know it's of IgG origin. Then now looking at the light chain subtractions, you see that there's 2/3 subtractions in kappa, and then there is 1/3 subtraction in lambda. So there's partial subtraction for both of the light chains. So obviously this is a mixture. It's an increased amount of IgG, but it's a mixture. It's not a monoclonality. All right. So doing the immunotyping also clears our doubt and fears with respect. Okay. So it's a polyclonal increase of IgG. Okay. So now we can move faster with this understanding. If we look at the monoclonal peak it happens in gamma fraction. Okay. So there is complete subtraction in the IgG, and there's only complete subtraction in the kappa. There isn't anything in the lambda. Of course, this peak is narrower with a pointed top. So clearly, this is an example of IgG kappa, right? So now let's look at the polyclonal increase of IgA. Right. So in this situation, in polyclonal increase of IgA, the abnormality is that there's higher beta 2 compared to beta 1. Okay, and this is not normal because the other should be higher. And we can see that there's elevation in the inflammation, acute phase proteins alpha 1 and alpha 2. So we zoom into the abnormality and look at how the change happens. You can see that there is subtraction in the IgG, but this will be because there are always IgG antibodies that are present. Okay, and then if you look at the IgA window, you can also see that there's a subtraction, and importantly, there's a bridge aspect between beta 2 and the gamma region, and the elevation of beta 2 occurs as a broad wave. So remember, the same principle applies. Even in IgA, you can see that the antibody increase has a broad wave. Remember, polyclonal is always a broad wave and a rounded top. Okay, so the same principle applies even though the IgA curve looks a little bit different. Okay, and then you look at the light chains. How are the subtractions happening? You can see that there is about 50-50. So for IgA, it's not the 2/3 kappa, 1/3 lambda. Okay, it's a 50-50. All right, so that's how we tell that this is not monoclonality but polyclonal. All right, and why is the why do we mark C3? If you look at the IgA curve, when you do the subtraction, you do not see the whole curve disappearing completely. The reason is because this is beta 2. Remember, in your beta 2 region, there are other proteins that are present. For example, complement C3 proteins, and these proteins cannot be removed by your anti-serial. Okay, it's only in the gamma portion that you tend to see very complete subtractions because most of the presence within it are the antibodies. But in the other peaks earlier on, like alpha 1 to your beta 2, these have other proteins that cannot be removed away by the anti-serial. Okay, so this is something that should be on mind to understand why they are not subtracted. Okay, we look at reduction of abnormalities, but whatever exists there that cannot be subtracted by the anti-serial remains. Okay. So conclusion for that is the polyclonal IgA. So if you look at the polyclonal IgA, remember in the abnormality would be the beta 2 that's higher. But look at the beta 2 now. Okay, number one, there's no bridge aspect between beta 2 and the gamma portion. It's like a deeper valley. Okay, and if you look at the IgA window, you can see there's a narrow reef and a pointed top. And when you look at the light chain subtractions, clearly it's not partial subtraction. There's more complete subtraction in your kappa. All right, so this is an example of, okay, so this is an example of a monoclonal IgA kappa, right? So now let's consider for IgM. If we have a polyclonal increase, remember I said that the IgM presence is more at the left side of the gamma portion. So here you see like a faint restriction in the left side of the gamma portion. There seems to be a little bit of elevation. All right, when we zoom in onto this, if you look at every curve that's present here, you notice that there's a bit of the subtraction in IgM. So if you look at all the blue color curves, the residual curves in every panel, you will notice that for all the gamma portions are more elevated on the left side. Okay, and only one would be IgM window where the left side is lower than the right side. So you know that a subtraction has happened in the IgM window, right? Then also importantly, it is to look at, so you see, for example, IgG, the rounded side remains there. Okay, the same rounded side is subtracted away in the IgM window. And then looking at the kappa and the lambda side to see is there like a complete subtraction in one and not the other, there isn't. Okay, so it's a partial subtraction. So this is the example of polyclonal IgM. So if you look at the monoclonal IgM peak, okay, it is a greater distortion of the left side of the gamma fraction. We magnify it, and then we look at where this distortion disappears. Okay, clearly it disappears in IgM. And if you look at where the distortion disappears more completely, clearly it disappears more completely in the kappa fraction. Okay, so complete subtraction in both the IgM and the kappa fraction. So we know that this is a conclusion of a monoclonal IgM kappa, right? So let's run through some integration exercises. So in this case, you see an elevated increase of alpha-2. Okay, there's no information on nephrotic context. Okay, so once again, zoom in on where the abnormality is and see where the subtraction happens. So it happens in IgA and it happens on kappa. So obviously it's the IgA kappa. Okay, I want to re-emphasize again, the underlying peak is not subtracted because out of the gamma fraction, everything that's out of the gamma fraction has proteins that are not antibodies within them. And the anti-serial cannot remove away these proteins, so the peak remains. Okay. So in this case, beta 2 is higher than beta 1. Again, without any inflammatory context, APAC disease. So once again, zoom in on the abnormality, and then we see where is the abnormality being corrected. It's being corrected in IgA, and it's being corrected in kappa. Okay, so clearly, again, the underlying band is not removed by the anti-serial because it's probably C3 complement in your beta 2. Okay, but you see a very complete subtraction of that abnormality. Okay, so it is the IgA kappa in this case. So in this case, you see a strong increase in beta 1 plus hypogammaglobulinemia. Okay, so again, zoom into the beta 1, and then you see where is it being corrected? It's being corrected in the IgA window, it's being corrected in the L. So obviously, this is the case of IgA lambda. All right, so in this example, you see that beta 2 is higher than beta 1, but as a whole gamma fraction is very thick. And it seems like there is a lot of antibodies in that region. All right, so once again, zoom in on the abnormality. And then after that, this case, I like you guys to see this case because if you look at the subtraction, you can see that in IgG, there is a complete subtraction in IgG. There is also partial subtractions in the lambda and the kappa in the blue arrows. Okay, so those will be the IgG-related subtractions. Okay, the whole fraction there is increased, but the subtraction is partial, so that we know that there's a polyclonal background, there's a polyclonal IgG increase. Okay, for the IgA, we see a subtraction there. It is a very broad-wreathed IgA fraction, but we only see partial subtractions for the kappa and the lambda in the yellow arrows. Okay, so clearly this is a mixture of polyclonal increase of IgG and IgA. It's not truly monoclonality. Okay, how about this case? You see a very small peak in the gamma fraction. Okay, then zoom in again, because when you have very small distortions, you can't really see it except with magnification. All right, so this is important to see where the subtraction happens. You only see the subtraction happening in the lambda window and not really in the other panels. All right. So in this case, it's a possibility that it's a monoclonal free light chain increase. But then again, because we did not look at IgD or IgE, which are much rarer in the disease, it's important for us to rule out the possibility that it could be IgD or IgE. So the way to do it is to do immunofixation, where you can use the anti-serial against IgD or IgE. There is also an anti-serial that goes against free light chain. And it will allow you to accurately detect that whether this is a free light chain or is it a light chain that's bound to a heavy chain. Okay, so with such, right, I will conclude the webinar and I would welcome you to pose your questions. I will invite you to support us by filling up the forms as well. For now, I do not see any questions in the questions box or the chat box. Please feel free to write your questions. And feel free to send in your questions to me even after this webinar. Okay, I see a question coming in. Oh, no, there's a comment. Thank you. Thank you. Okay, I do not see any further questions from this, but as such, I would like to thank you all for attending this webinar today. And please feel free to reach out to me if you have further questions to this. If you need more presentations or clarifications on it, just feel free to reach out as well. Okay, so thank you. Thank you. I see the comments. Thank you very much for attending, and hope to see you guys soon again. Take care. Goodbye.